Morning, everyone. Thank you for your valuable time. I'm Hojin Cheon, and I'm currently a master's student at Korea University in South Korea. Today, I'm going to talk about my research, Cellytics digital inline holography platform. Here are the contents of this presentation. Analyzing the cells and vertebrate is important in disease research or marine pollution measurement. That's why we developed Cellytics. It's a platform based on digital inline holography, which offers a faster, more accurate, and cost-effective way to analyze the cell without extensive equipment or technical expertise. Before we get deeper into Cellytics, I'd like to briefly explain the technology we use, the lens-free shadow imaging technology. We call briefly LSIT. This method uses semi-coherent light of specific wavelengths to illuminate the cells and capture the shadow images without any lenses. The difference in refractive index between the inside and outside of the cell create the shadow diffraction pattern. Through the amplitude information, we can determine the cell size and internal characteristic. And these shadow images are analyzed using a cell detection algorithm that extract the detailed information about the cell, including a size, a shape, and internal structure. Using LSIT, we developed the Cellytics platform. What's remarkable about Cellytics is its small size, low cost, and portability. It's possible to rapid analysis in under a minute, and it's user-friendly. When we capture the shadow images of cells using LSIT, we obtain the data like the graph shown here. Draw a line in a circle at 15 degrees intervals on individual shadow images, and measure the gray value and to calculate the average value and standard deviation values. Based on the characteristic of this graph, we designed various shadow parameters. For example, PPD is related to the size of the cell, and MMD is related to external complexity of the cell. And WSM-SD or WSM-STD is the same word. Anyway, so WSM-SD related to the internal complexity of the cells. And we separate human blood into red blood cell, RBCs, and white blood cell, WBCs, and capture their shadow image using LSIT. As expected, the shadow image of RBCs and WBCs were noticeably different. And also, we prepared the microsphere with diameter of 10 micrometer and 20 micrometer, and capture their shadow images using Cellytics. Then these shadow images were analyzed using a control algorithm. We also tested different mixing ratios such as 3:7, 5:5, and 8:2. And also we analyzed the locomotion of lotipers from recorded video using machine learning. And lotipers are commonly used in ecotoxicology and physiology studies. For this experiment, we used the toluene and acetone as the toxic substance. In addition, various experiments were conducted based on the characteristic of cells and disease. Now is the result from various experiments. Through the principal component analysis, we were able to clearly distinguish it between 10 micrometer and 20 micrometer microsphere by plotting a two-dimension scatter plot. We confirmed that the two groups were well separated. And for blood cell, we matched the shadow images and with the optical microscope images, and used the heat map of shadow parameters to clearly separate RBCs from WBCs. This demonstrates the potential of Cellytics in cellular classification. And this slide compares the traditional immune cell activation assays with the Cellytics platform. In traditional method, it takes 24 to 72 hours to analyze cell proliferation or cytokine secretion. And the process is quite complex. But on the other hand, with Cellytics platform, we can analyze the cell activity in under 5 minutes and just using a 10 microliter of a sample. This new approach is much faster and more efficient compared to the traditional method, significantly reducing the time. And it offers great potential for both research and clinical application. The CDC cross-matching is tested to determine whether organ donation is possible between a donor and a recipient. The traditional method involves staining cells with fluorescent dyes to assess cell death, which helps evaluate the feasibility of organ transplantation. However, this process is invasive and can be subject to expert bias. The graph at the bottom compares the result from Cellytics and traditional fluorescence microscopy. The correlation between the two methods is extremely high, and demonstrating that the Cellytics can achieve accuracy comparable to manual microscopy. In fact, the correlation index is more than 90% all range. In conclusion, Cellytics provides a fast and reliable method for analyzing cells viability and showing a great potential as an alternative to conventional method. And in addition, we also applied Cellytics for environmental toxicity analysis using lotipers. We exposed the lotipers to toxic substances such as toluene and acetone, and used Cellytics to measure their real-time locomotion. Then machine learning algorithm were applied to analyze the movement, allowing us to assess the impact of these toxins. Now, this is a summary. The simple algorithm classifies micro-sized particle with significant difference. For the improvement will enhance classification, and a significant difference between groups were founded. Future studies may classify white blood cell subgroups. And in locomotion of Lotifer, real-time monitoring of marine pollution is feasible and will be tested for various toxic substances. Thank you so much for your attention.